Kshitija, a DM medical geneticist from AIIMS New Delhi, currently working as a consultant clinical geneticist at Apollo Hospitals, Navi, Mumbai. On behalf of Organization of Rare Diseases India, ORDI, I welcome you to yet another knowledge-packed webinar of the Rare Info series. ORDI has been doing commendable work in raising awareness about rare disorders and creating this invaluable platform which bridges doctors, patients, and their families. Such initiatives foster meaningful discussions, allowing us to understand rare disorders from both medical and patient perspective, an essential step towards better care and support. These webinars are recorded and are available for viewing on their YouTube channel, which goes by the same name, that is Organization for Rare Diseases India, ORDI. Coming to today's topic, in recent years, genetic testing has become increasingly accessible. Yet, there remains a considerable confusion. Which test to choose from this wide array of tests available? What are the scope and limitations of each? To help simplify these complexities, we are privileged to have with us an expert in this field, Lieutenant Colonel Dr. Shilpa Jauhari. Dr. Jauhari is a pediatrician trained in genetics, holding a DNB pediatrics and a 6-month specialization in clinical genetics from Center for Human Genetics. She is currently serving as an assistant professor of pediatrics at Command Hospital, Air Force, Bangalore. Let's welcome Dr. Shilpa as she guides us through the nuances of these advanced genetic tests in her talk, which is aptly titled Blueprint Breakdown, Intro to Genetic Testing. A gentle reminder to the participants for typing any questions or queries that they have in the Q&A section. These questions will be addressed at the end of the talk. Over to you, Dr. Shilpa.
Thank you so much, ma'am. Am I audible, ma'am?
Yes, yes.
Okay.
Please go ahead.
So today we are going to see what genetic testing is all about and how can we do the best out of a genetic test. And let's start. So can we have the next slide, please? So aptly it is said that the genome is the book of life. Every letter tells a story of our past, present, and future, guiding medicine to heal across all specialties. Next slide, please. Genetic disorders are not rare. So, as was a thought earlier that they are very rare, it's not so. In our OPD full of patients, out of 20 patients, one person would be linked to a genetic disorder. There are more than 7,000 known Mendelian disorders, and early suspicion and accurate diagnosis improves the outcomes. This is a well known fact. Multidisciplinary collaboration is the key for holistic care. This is also well known. So, we start with the famous analogy of the genome with the book of life. We can compare the genome with the book of life. Next slide. And DNA as the text. DNA is the fundamental molecule carrying genetic information. It's composed of a sequence of chemical bases, which are A, T, G, C, which can be compared to the letters in a book. Next slide. Genes can be compared to sentences, where genes are specific sequences of DNA that codes for proteins or functional RNA molecules. They are like sentences in the book that convey specific instructions for making a specific thing. Chromosomes can be compared to chapters in a book. They are structures that organize and package DNA into a manageable form. They are analogous to the chapters in a book, grouping related genes together. Next slide. And mutations can be compared to spelling errors. Genetic mutations are changes in the DNA sequences. These changes can be compared to spelling errors in a book, which may or may not alter the meaning or function of the sentence or gene. Next slide. Insertion and deletions can be compared as missing or extra words. Insertions of or deletions of DNA sequences are like adding or removing words from the book, which can significantly impact the overall meaning or function. Next slide. Genetic testing is like analyzing this book. It involves examining the DNA sequences to identify specific mutations or variations. It's like carefully reading the book to understand its content and identify any errors or unusual features. Next slide. So, what's the need for genetic testing? Yes, there's a need for genetic testing to form a definitive diagnosis. Why? To have the management protocols, prognostication, and natural history of the disease. It's important for genetic counseling, prenatal diagnosis, and for carrier testing. Next slide. Attributes of a test. A test for a doctor and a patient holds importance. The doctor must know before ordering a test what is the principle of the test, the indications, the interpretation of results, and the limitations. And the patient should be told about the need for the test, the benefits, and limitations. Next slide. So, just briefly about the timeline of advancements in genetic testing. In 1953, Watson and Crick described DNA's double helix structure. In 1977, Sanger sequencing was done. 1983, PCR was invented. 1986, first genetic test for cystic fibrosis identified specific gene mutations. In 1990, the famous Human Genome Project began. 2003, it was completed. Next slide. 2007, NGS emerged. 2013, CRISPR-Cas9 gene editing was introduced, enhancing the targeted gene testing. In 2018, whole genome sequencing became more affordable. In 2022, polygenic risk scores improved, disease prediction using multiple gene variants. And in 2025, AI has also come in. Next slide, please. So these are certain, this is a diagram showing genetic disorders and what type of disorders are there and how we go about in identifying them. So if we go from the cell, there can be structural chromosomal aberrations in the chromosomes. Then if we go further, there can be copy number variations. There can be monogenic disorders, or there can be point mutations. In the genetic disorders, first in the chromosomal ones, we can also have aneuploidies, which can be detected by various tests, which we'll be dealing with later. Next slide, please. So genetic disorders, as we said, in this slide, we just reiterate, they can be chromosomal disorders, microdeletion duplication, single gene disorders, triplet repeat disorders, mitochondrial and epigenetic disorders like imprinting disorders. Next slide. Genetic testing is analysis of human DNA in any form, any of its form, or related products like chromosomes, RNA, or proteins. And there can be three basic type of tests like cytogenetic test, molecular test, and biochemical test. Next slide. In this presentation, we'll be dealing with the different types of genetic test, their indications, principles, advantages, and limitations. So first, we'll be dealing with cytogenetic testing. To know about cytogenetic testing, we first should know about the type of disorders we are screening them for, we are identifying them with. So, chromosomal disorders can be two types, numerical abnormalities or structural abnormalities. Numerical abnormalities can further be grouped into aneuploidies like trisomy, monosomy, or polyploidy. Structural abnormalities can be grouped into deletion, duplication, translocations, pericentric or paracentric inversions, ring chromosome identification, or isochromosome identification. Next slide. So the first test we'll be talking about today evening is karyotype. So first we start with the indications. The indications of a karyotype are if we suspect some chromosomal abnormalities, any unexplained intellectual disability in a child, disorders of sexual development, or we see a patient in the OPD with ambiguous genitalia, we see a patient with short stature, especially prepubertal females, where we are suspecting Turner syndromes. Then in a gynae OPD, we can come across patients who are coming up with recurrent pregnancy losses, infertility issues, parents of a child with some structural chromosomal abnormality and malignancies. Next slide. So this is the structure of a chromosome, and there are different types of chromosomes like metacentric, submetacentric, acrocentric, depending on the position of the centromere, and it has got two arms, a short arm and a long arm. Next slide. This is just a pictorial representation of a G-banded metaphase spread. Next slide. And this is how a conventional karyotype looks like. You can see that in this it's a karyogram where the chromosomes are arranged in according to the size, and the smallest size is the Y chromosome, and the sex chromosome as a dictum, they are represented at the end, whereas the others are shown in pairs and size wise they are arranged. Next slide. So how do we do sampling for karyotype? In while sending a sample for a karyotype, we have to keep certain things in our mind which are very important, because if we do not follow them, then the sample might go bad and the culture might not grow. So we have to take a venous blood. We have to send 2 ml of blood in a heparin vacutainer, and the storage at 2 to 4 degrees centigrade. Transport is preferably within 24 hours. We can also do karyotyping in a prenatal sample, and we'll come to about it later in the later slides. Then preparation of a karyotype. This is a diagram depicting how a karyotype is prepared. Metaphase spread is done and then they are read. Next slide. So these are certain symbols used in describing a karyotype. The P represents the short arm, Q the long arm, centromere, deletion, duplication, fragile sites, isochromosome, inversions. This is how we have to write the chromosome report, the karyotype report if it comes and has a certain anomaly. Next slide. This is a karyotype which is showing triploidy. Next slide. So this is translocation. The above one is Robertsonian translocation. Below you can see reciprocal translocations. Next slide. Karyotype is the oldest technique for genetic testing. It's a visual assessment which is done under the microscope. Whole genome can be seen in one go, and it can detect balanced rearrangements, which is a very important part of this test. Can detect location of extra chromosome material also. Next slide. But however, there are certain limitations of karyotype because it has a low resolution. Chromosomal rearrangements greater than 5 MB size can only be detected. Hence, microdeletion syndromes like Williams can have a high probability of getting missed in this. Test needs actively dividing live cells. So the sample has to be sent as soon as possible to the lab. It's labor intensive. Turnaround time is 15 days. Abnormalities in telomeric region are difficult to identify. Next slide. So this is a case scenario where there's a couple with 4 first trimester abortions, and the karyotype which was done for them showed a 46XY translocation 4 and 15. There is a translocation between chromosome 4 and 15, as you can see in this diagram with red marked with red arrows. So this is how we came to this diagnosis of a couple with 4 first trimester abortions. Next slide, please. So the next test we come to is FISH, which is known as fluorescent in situ hybridization. And the principle for FISH is it's a complementary hybridization where fluorescently labeled oligonucleotide probes are used for hybridization with metaphase chromosomes or interphase cells. Very high number of cells can be evaluated for low level of mosaicism. This is one of the advantages of FISH. And the other most important advantage is that FISH gives quick results. The turnaround time is around 72 hours for this. Next slide, please. So what are the indications? When will you send a FISH? When clinically suspected chromosomal microdeletions like DeGeorge, Williams, Creduchat, Prader-Willi, prenatal FISH for chromosomal aneuploidies like trisomy 13, 18, 21, and sex chromosome aneuploidies, cancer genetics like BCR-ABL, pre-implantation genetic diagnosis. Next slide, please. These are various probes which are used for different microdeletion syndromes, which I wanted to show because this is important because uh, we can fish out only if we know the diagnosis and then we send the fish. If we send a fish test for some disorder which we are not sure of, we tend to miss certain disorders in this. Next slide, please. So this is a case scenario where a 2-year-old baby boy with cleft palate, small ASD, hypocalcemia, recurrent infections. Clinically, it was suspected that the baby was suffering from DeGeorge syndrome. The clinician ordered FISH to look for 22q microdeletion, and it came out to be positive. So how do we interpret it? Next slide. Yeah, so in this diagram, we can see that the normal fish has got There are two colors you can see in the slide: green, and two red. However, in the next diagram, you see after the arrows, you see that there is only one red and two green. That means that the specific probe for 22q microdeletion was used, and there was a deletion which was found, because of which the fluorescence was absent. Next slide, please. So, uses and advantages of FISH are: it detects known microdeletions and fast reporting. Limitations, however, are: clear clinical suspicion is essential to order FISH, cannot be used for unknown microdeletions, it's labor intensive, and resolution is 300 kb to 1 MB. Next slide, please. So, coming to end of cytogenetics, karyotyping and FISH are essential cytogenetic tools in pediatric and clinical practice. It's useful for detecting numerical and structural chromosomal abnormalities, the karyotype. FISH provides rapid, targeted detection of specific genetic abnormalities, and appropriate test selection is crucial for accurate diagnosis, prognosis, and management of genetic disorders. Next slide, please. Now we come to, after cytogenetic testing, we come to molecular tests, which are DNA-based tests. So the principle of DNA-based test is mainly to either complementary hybridization or polymerase chain reaction. Next slide, please. So the complementary hybridization example used in southern blot, dot blot, or microarray is based on the fact that the adenine will pair with guanine ATGC. A will pair with T and G will pair with C. Next slide, please. So polymerase chain reaction, it's a DNA cloning method. There is a chain reaction because of which there is a polymerization which is taking place. This technique takes a specific sequence of DNA of small amounts, and later on it amplifies it to be used for further testing. It is rapid, extremely sensitive, and robust test. It uses templates, primers, polymerase, nucleotides, buffers, cofactors, and the tests which are based on this are tests like NGS, DNA sequencing, PCR, and RFLP. Next slide, please. Quantitative PCR, quantitative fluorescent PCR, QF-PCR, which is called in short, is a rapid molecular technique using fluorescent labeled primers to amplify short tandem repeat markers. It's a PCR-based technique, and technically it is easier than FISH. It is less costly, and prenatal diagnosis of aneuploidies can be done when time is very important, very crucial, for aneuploidies of chromosome 13, 18, 21, and sex chromosomes. Next slide, please. So QFPCR detects aneuploidies and by DNA dosage. It's a faster alternative to karyotyping, and it provides results within 24 to 48 hours, even faster than FISH. And it's rapid, highly sensitive, and cost effective for common aneuploidies, and requires only small amount of DNA from amniotic fluid, CVS, or blood. Next slide, please. However, this test again has certain limitations, like it cannot detect structural chromosomal abnormalities, example balance translocations or microdeletions, and it is limited to targeted aneuploidies and not a substitute for full karyotype or microarray. Next slide, please. So this is a diagram depicting QFPCR. Next slide, please. This is the structure of a gene, how a gene looks like. This is example of a small gene. For example, this is a HBB gene, which is a 3-exon gene, which has got exons, 3 exons, and in between there are intronic regions. There is a translation initiation codon, transcription initiation is there, promoter region is there, and then there is at the end polyadenylation signal. There is a 5' end in the beginning and the 3' end in the at the end. Next slide, please. So this is a diagram depicting central dogma that DNA to RNA to proteins is the main mechanism by which proteins are formed, and how we can go about testing at these different levels. Next slide, please. So this is just a diagram showing structure of a DNA, which is made up of a pentose sugar, a phosphate group, and a carbon-nitrogen ring structure, which is called a nitrogenous base. Next slide, please. So human genome variations, we know that the genome is 99.5% identical between any two unrelated humans. And there are several genetically determined differences among individuals which can be there. change of a single nucleotide to alterations of an entire chromosome can be present. However, most changes impact various traits. They are benign variants, but there are few changes which alter normal protein and thus bodily functions, and they are known as pathogenic variants. Next slide. So, what is polymorphism? If polymorphism is a change, the frequency of which in a population is more than 1% of the alleles, it is a benign change. Usually, it is inconsequential to the functioning of any gene and can lead to variant proteins if located in exons, resulting in different traits. Next slide, please. This is the pathogenic variations. They can be chromosomal rearrangements, copy number variations, more than 50 base pairs, indels, which are insertions or deletions. Then single nucleotide variants can be there, like substitutions, deletions, insertions. Next slide, please. CMA is chromosomal microarray, and it's a microarray based cytogenetic analysis. The principle is base pairing of A, T, and GC. It's hybridization based. And in this test, there are about 750,000 chips which are used, 550,000 unique non polymorphic probes, and approximately 2 lakh SNPs are used. Resolution is about 100 kb, and deletion duplication can be found out if they are present anywhere in the genome. Next slide, please. However, again, this test has got certain limitations, like it identifies only copy number variants, that is, gain or losses, along with the regions of heterozygosity. Any other form of polyploidy, truly balanced chromosomal rearrangements like inversions, balanced chromosomal imbalances, point mutations, small deletions, or mosaic conditions cannot be detected by this chromosomal microarray testing. Next slide. This is a report of a patient where we are seeing that this is CMA. This is report in which no significant copy number variations were detected. And this is how a test report is seen, which is according to the ISCN nomenclature. Next slide, please. The next test we come to is MLPA, which is multiplex dependent probe amplification, which has got certain steps like denaturation, hybridization, ligation, amplification by PCR, fragment separation, data analysis is the last test. In the subsequent slides, there are diagrams depicting this test, which we'll just rush through. Please go ahead with the next slide, please. So this is MLPA, the company which gives the holds the patent for this MLPA testing is MRC Holland. Next slide, please. This is a MLPA test. This is a test showing a normal copy number diploid gene, where we see in the reference samples and in the test sample there is no aberration which is detected. Next slide. So coming to the next, where there is a heterozygous deletion, we see the reference sample and the test sample. In the test sample, you can see 2 red dots below, which show a heterozygous deletion. Next slide, please. And this is a duplication, heterozygous duplication, where you can see in the second diagram that there is a heterozygous duplication where those red dots are seen in the above part. Next slide, please. So these are MLPA probe oligonucleotides, which the sequence of the test will be shown in the later slides. Next slide, please. So there is, after the step, it is sample denaturation and probe hybridization which occurs. Next slide. And then the next thing which happens is probe ligation. Next slide. And then after the probe ligation, we have probe amplification, where amplicons are formed. Next slide. And then fragment separation by capillary electrophoresis is seen. Next slide. And finally, we have data analysis, where we come to know if there is any deletion or duplication. Next slide. So indications for doing this test is detection of aneuploidies of chromosomes 13, 18, 21, XY, detection of exon level deletion duplications like we do commonly in diseases like DMD and SMA, detection of large chromosomal deletions or duplications like in DeJoy syndrome, Williams, or subtelomeric regions, and detection of gains and losses of genes in cancer tissues. Next slide, please. Again, like other tests, this test also has got certain limitations, Like it can detect only known deletions, duplications. Up to 40 fragments or exons can be checked at a time. And single exon deletion should be confirmed always with another method. Next slide, please. This is example of a MLPA report, how it looks like. So in this, you can see that this is a carrier or pathogenic variant causative of a suspected phenotype. The phenotype was DMD. Like features, so it is a heterozygous deletion in exon 1, and the MLPA probe ratio is 0.53, which is coding for Becker's muscular dystrophy oblique DMD. It's a X-linked disorder and pathogenic. Next slide. So what is MLPA ratio? MLPA ratios or dosage quotient, they are ratios if they are below 0.7 or above 1.3, they are indicative of a deletion, copy number change from 2 to 1, or duplication, that is copy number change from 2 to 3 respectively. A dosage quotient of 0.0 indicates a homozygous or hemizygous deletion. 0.35 to 0.65 means heterozygous deletion. 1.35 to 1.55 indicates heterozygous duplication. 1.7 to 2.2 indicates homozygous or hemizygous duplication. And MLPA ratio between 0.8 to 1.2 indicates a normal copy number status. Next slide, please. This is a report showing MLPA of a DMD patient where we can see a deletion. Next slide, please. And apart from MLPA, we also have testing like MS-MLPA, which is methylation specific MLPA, which are done for methylation disorders. So it is caused by abnormal DNA methylation affecting gene expression, often without DNA sequence change. So the example of these disorders are Prader-Willi, Angelman, Beckwith-Widman. Next slide. Apart from this, there are triple primed PCR testing, which is a PCR based method used to detect and characterize expanded triplet repeat sequences uh, like CGG or CAG repeats, often found in human genetic diseases. It's particularly useful for identifying large expansions that can be missed by traditional PCR methods. In this diagram, we can see that the PCR findings of a FMR1 gene. Next slide, please. So this is the report of a Fragile X male, where we can see that there are increased peaks and triple prime PCR. There are repeats more than 200. Then it will be affected male. Next slide, please. Next, we come to ACMG guidelines. ACMG guidelines are internationally accepted guidelines for the interpretation of variants, and they are updated in 2015. The current version has been the standard for terminology and methods as the scope of clinical genetic testing has gradually diversified with the development of sequencing technology. ACMG guidelines, they were established by clinicians, clinical lab directors of ACMG, that is American College of Medical Genetics and Genomics, AMP, that is Association of Molecular Pathology, and CAP, that is College of American Pathologists. Next slide, please. So this is how ACMG classifies the various variants. There are five classifications. It can be a variant if found in a test. It can be classified as pathogenic, which is disease causing, likely pathogenic, variant of uncertain significance, likely benign or benign. Next slide, please. So this is a Sanger sequencing, which was developed by Sir Frederick Sanger in 1977. It's also known as dideoxy chain termination method, and the principle it uses is the use of dideoxynucleotides for DNA synthesis. The synthesized strand sizes are determined for each dideoxynucleotide reaction by using gel or capillary electrophoresis at the end. Next slide, please. So this is a case scenario where we use Sanger sequencing. There is a male who comes to you in OPD with short stature, frontal bossing, depressed nasal bridge, mid-face retrusion, rhizomelia, and you send for a Sanger sequencing suspecting achondroplasia, and you find this report where there's a variant in the C.1138G2A, which is one of the most common variants found in this disorder. Next slide, please. Okay, in the FGFR3 gene. So this is NGS next generation sequencing. It can be, we can send a clinical exome sequencing, whole exome sequencing, or a whole genome sequencing, where there is a reference genome, and we try to find out variants in that. So clinical exome sequencing, what it is, we we sequence the exomes which are known to be causing diseases which are already known. Around 5,000 disorders, 5,000 genes can be uh, exomed in this. Whereas in whole exome sequencing, we sequence the whole exome, uh, which can give us new or novel variants also. And in a whole genome sequencing, we will be, in addition to the exons, we'll be sequencing the introns also. Next slide, please. So, what are the indications of exome sequencing? If we suspect, we take proper history, evaluation, examination, investigations are done, and a proper pedigree is drawn, and we find that it looks like a Mendelian disorder, Mendelian genetic disorder. We can send exome sequencing, whole exome sequencing, called as WES for short. Single gene candidates are eliminated. Phenotype known to be genetically heterogeneous. Expensive multi-gene testing is there, then we can send. Or there is unknown genetic etiology, or there is a diagnostic odyssey, then also we can send a WES. Next slide. So, what happens if we send a WES and the report comes? There are three possibilities. Either there is a potential disease causing variant which is identified, or there is no disease causing mutation found, or there are certain incidental findings which are found. So, what do we do? If a potential disease causing variant is identified, then we know that the phenotype and genotype is matching. Then we say that yes, this is the answer, and we can segregate in the parents also. Next is, if there is no, last slide please. Yeah. So no disease causing mutation is found, then we have to reassess what went wrong. Was the inputs, the phenotypes given correctly? Uh, was the test the right choice of test, or something got missed in the way? Because generally, if we give the inputs properly, we do the examination properly, the phenotyping is correct, we tend to get an answer. Incidental findings are something which can be there, which are not matching with the phenotype. However, the lab finds them, so then there's a method by which we can do reverse phenotyping and check whether this fits in or not. Next slide, please. So what does VOUS means to a clinician? VOUS means, that is variant of uncertain significance, that we have to review the phenotype and order more tests to confirm or rule out a diagnosis. We have to review the literature, do our own assessment again, review literature and phenotype again at a later date, or reanalysis at 6 to 12 months time, or additional monitoring of the patient for the disorder in question can be done. Next slide. And one thing which is missing in this, which we can also do, is we can segregate the variant in the parents. If we feel strongly that it's a VOUS and the patient, the genotype phenotype is matching, we can segregate in the parents also. So this is a recent publication from ACMG practice guidelines, where they say that they strongly recommend exome sequencing and genome sequencing as a first tier or second tier test, guided by clinical judgment and often clinician patient family shared decision making after CMA or focus testing for patients with one or more congenital anomalies prior to 1 year of age, or for patients with developmental delay or intellectual disability with onset prior to 18 years of age. Next slide. Like other tests, this test also has a limitation, and the limitations include inability to diagnose repeat disorders. Like if we feel a patient coming to the OPD is having intellectual disability, long face, other features of fragile X, and we order a VES, we might tend to miss the diagnosis. Also, if there is a patient suspected of Angelman and we send VES, we might tend to miss it in the whole exome sequencing, where we'll have to do a MSMLPA for the phenotype. In addition, exome sequencing may have poor coverage of certain genomic regions, reduced sensitivity at detecting intragenic deletions, especially those less than 3 exons in size, and will not detect balanced chromosomal abnormalities, especially translocations. It is also important to note that the lab interpretation of exome or genome is very much dependent on what we tell the lab. What is the phenotype input which has been given? to the lab by the ordering clinician. Hence, that is of utmost importance. Next slide, please. So this is a slide which is showing that in the coming future there might be a time when we'll go for maybe for whole genome sequencing if the prices come down further. They have already come down. So there are certain advantages, there are certain disadvantages of it compared to exome sequencing. So the main advantage is that it is a faster library preparation is there. It includes introns, includes regulatory elements. A better detection of copy number variant CNVs can be detected in whole genome sequencing. Detection of structural variants can be done. However, there are certain disadvantages like it's of greater cost. The sequencing costs more. Greater cost of data storage also because it's a huge amount of data. Many more variants are there to analyze and difficulty in interpreting non-coding variants. Next slide. So basically, by testing, by phenotyping and genotyping the patient, we are searching for what went wrong and what can be corrected. So if there are therapies in future, then we can we should always try to find a cause so that at least we have a known cohort which may benefit, if not now, maybe in the future. Next slide. And last but not the least, the most important thing before and after ordering a test is genetic counseling. We have to have a written um, documentation of the pre test counseling before ordering the test. When we tell the patient that why the test is important, what will be the turnaround time, what will be the approximate cost, what all can be found in the test, what implications it might have, and then when the test comes, you have to tell them that you'll call them for a post-test counseling in which you have to explain, you have to sit with the patient, preferably with the test report in hand. If not, then on the soft copy maybe. But you have to show them what each and everything means so that they get it. Many a times patients come to us in the OPD saying that a genetic test was done, but they don't know what came out of it. It was normal, and then later we found that there was a heterozygous variant which was present, and no one explained them that what implications it might have in the future. So this is really very important. So next slide, please. So there might be more tests which might come in the future, which may there might be one test detecting all the things, but as of now, for different disorders, there are different testing methodologies which we have to follow, and according to the phenotype of the patient, we have to order. Hence, phenotyping, reading about the disease is of is very important. Next slide, please. I would like to end this class by reiterating the words of Haruki Murakami, who says that human beings are ultimately nothing but carriers, passageways for genes. They ride us into the ground like racehorses. From generation to generation, genes don't think about what contributes good or evil. They don't care whether we are happy or unhappy. We are just means to an end for them. The only thing they think about is what is most efficient for them. Thank you.
Wonderful, wonderful presentation, Dr. Shilpa.
Thank you, ma'am.
I think you covered everything it's actually a very comprehensive topic with so many different, subsections of genetic testings, and you covered it so well. And I love the analogy of the book that you gave in the beginning for easy understanding. So that was excellent. And so among the initial tests, you mentioned about these importance of sampling and proper storage while sending over. So, I think among these most fragile are these karyotype samples, because as you said, you need to grow the cells. So, sometimes we send these products of conception for testing, and karyotype many times there's a high rate of failure. So in that case, what can be our options, like if karyotype fails?
Ma'am, one more most important thing I would like to tell everyone is that what I have learned is that
Mhm.
in a country like India, where if there's a abortion, people generally used to throw the product of conception away without taking
Mm-hmm.
any photographs, without doing any phenotyping, which is,
Uh-huh.
I should reiterate it again and again, that it's very important because the phenotype also tells us a lot. Second thing is DNA storage, which can be done. If the fish or the karyotype which was sent, that test doesn't turn out to be what we expected, or the
Mhm.
karyotype fails, then the DNA storage which was done, that can help us out in deciphering what actually
Okay,
it was.
right, right. So I think that thing should be kept in mind because otherwise, once that thing fails, then we don't have anything else left. So I
Right.
think DNA storage would be an excellent thing to do. Right. Excellent. So, and among the tests you mentioned about a TP-PCR testing. So, what are so as I understand, there is no blanket test for all disorders. I think it has to be evaluated clinically, and once depending on what disorder is being suspected, we have to send the test accordingly. So, what are some disorders where TP-PCR needs to be ordered?
Ma'am, triple prime PCR is generally for triple repeat disorders like Fragile X, where there are CGG
Okay.
repeats, then there are Huntington's chorea, there is
Mhm.
myotonic dystrophy, which can be there. Then there are also certain spinocerebral ataxias,
Okay.
which can be
Yeah.
seen by this. So there are multiple disorders which need
And
testing.
depending on what is suspected, I think the test will have
Or
to be
based on the
decided.
clinical suspicion and phenotype, we can send these tests.
Okay. Right, right. So that's what, and I think you very nicely explained the complexities of exome, analysis, like what the results that come and how they need to be interpreted. So I think so what would be some pointers while sending an exome sequencing, what information needs to be sent to the lab, or, how we can ensure that we are able to get proper finding what we are suspecting.
Yes, this is a very important question. I'm glad you asked this, ma'am, because I feel that many a times what notion we have, and even I had before joining this 6 months fellowship, was that if you send a genetic test, it will give you all the answers. So we just write that the patient is suffering from this, and we get the answer. But it is not so, and never will be so, because just like any other form of medicine we are practicing in genetics. You first, when a patient comes, we take a proper history, then we take a
Hmm.
proper examination, and then we do the relevant investigations, and then at the end we send the genetic testing. So whenever, especially in the case of dysmorphology, if we find dysmorphic features, I feel that the eyes see only what the mind knows. So in genetics,
Hmm.
I feel we have to keep on reading and reading, and we have to see the patients for experience. Both the things matter a lot, and especially in dysmorphic babies, we have to write down all the dysmorphic features we saw, or any MRI scans, imaging were done, the features which were present, the eyes, ears, anything, any finding which we find, we have to mention, so that the bioanalyst team in the labs, they get to
Hmm.
know that what is it that we are dealing with, because they will not have direct contact with the patient. It's only we who tell about the patient to them. So accordingly, they'll rule in or rule out the variants which are found.
Right, right. Rightly explained. Correct. So I think phenotyping is so important and providing the complete information, detailed information. Right. And also what you said about counseling the patients, I mean, post-test, of course, it's important, but I think pre-test is very important because probably, can you explain some scope of this pre-test, like what all we tell to the patient or what information we should give?
In the pre-test counseling, many a times patient come to us in the OPDs. They don't have the they are not knowing why this test needs to be done.
Mm.
The most common thing which I have heard is they are having one child who is suffering from
Mm.
any disorder, and they are not planning for the next pregnancy. And they say that people had told them to get a genetic testing done, but they said that in spite of the test, the child who is affected will not be benefited. So it's no use of sending the test, they say. But then we have to counsel them why it is important for the future
Mm.
generations. If they have a normal child, then also it is important for that child also in the future,
Mm.
plus for the carrier screening, for the other families in the extended family who might be
Mhm.
planning for babies, it might be important. So we have to explain them all the things. We have to
Right.
actually, in genetics, I feel we have to give more time to the patients and a regular OPD. So my
True, true.
run till the afternoon and they never finish off in time, ma'am.
Right, right. And again, we have to prepare them for any additional findings that might come,
Yes,
some incidental thing, whether
yes,
they want to know it or not. And if at all it, comes, then they should be
be
prepared
ready.
for it.
Yes, exactly, ma'am.
True. And I think VUSs are like a nightmare for
Yeah,
uh, doctors. And you told a very nice, bullet point wise way of how it is evaluated. So I think that was again, very nicely explained, because I think that is one thing which confuses a lot of
a lot in the OPDs,
doctors
ma'am.
who are not like working in core genetics. So that
And
was
many
great.
times it happens that we find follow-up patients whose a single test, genetic test was sent, a VOUS came, and that patient was
Mhm.
left. No further
Mhm.
testing was done, thinking that
Ah.
the genetic test has come and this is the report. Now nothing can't be done. So I want
Mm.
to say to all the people that no, if a VOUS has come, it means you have to search more. You have not got
Mm.
the answer. So we should not stop at that because it has got implications and very important implications, ma'am.
Right, right. And I think you very nicely explained, like given an overview of the ACMG classification, so that people understand what it means, like what is a VUS or what it is. I mean, so it's not like, a black and white report. There's so
Yeah,
much more to it.
ma'am.
So that was excellent. Very, very nice, and very like a very huge topic covered in a very nice, comprehensive, precise way with the right messaging. So that was excellent.
Thank you.
So I was just checking the Q&A section, so that there are no more questions as such. A lot of thumbs up and hearts coming our way. So that's great. Uh, Okay, excellent. So thank you so much, Dr. Shilpa, for this insightful and enlightening talk.
Thank
Your
you, ma'am.
expertise has provided the audience with a much needed clarity on genetic testings, helping us navigate its complexities with greater confidence. I would also like to express my thanks to ORDI for giving us this excellent platform. And finally, a big thank you to all our audience for their attentive participation and thoughtful engagement. Your presence and curiosity makes these discussions more impactful. Have a wonderful evening, everyone.
Thank you, ma'am. Good day, ma'am.